Hello, my name is Eric Gangloff from Ohio Wesleyan University, and I'm happy to offer this short video presentation on adaptation and plasticity in the multivariate thermal phenotype of common wall lizards. This is a project that was designed and conducted with Brooke Bodensteiner from Yale University, as well as our co-authors Martha Muñoz, also from Yale, and Laura Kouyoumdjian and Fabien Aubret from the CNRS in France. Let's start by talking a little bit about thermal phenotypes generally. First of all, we know that these phenotypes are multivariate. So here's an example of a thermal performance curve. You're probably familiar with this. This shows body temperature on the x-axis and some measure of organismal performance on the y-axis. From this curve, we can extract important parameters such as critical thermal minimum, critical thermal maximum, optimum temperature for performance. We can also describe selected and preferred body temperatures and performance breadth. Now, these curves can change in a number of ways. They can shift to the left or the right, or the shape of the curve can change without the endpoints necessarily moving. These changes can occur through processes of plasticity, such as acclimation, as well as through adaptation. However, these traits are not independent. They are linked through shared developmental pathways or through the effects of pleiotropy. Finally, we know that temperature interacts with other abiotic factors to influence organismal performance. So for example, here is a temperature-oxygen performance surface that describes the interactive effects of oxygen availability and temperature on organismal performance. It was within this background that we designed an experiment with the common wall lizard, Podarcis moralis. This is a widespread species found across much of southern Europe, generally at low elevations. However, in recent years, as temperatures have warmed, this species has been observed moving up in elevation to track these preferred thermal environments. As these organisms and many others move up in elevation, however, they face additional constraints, specifically the reduced oxygen availability at high elevations. So, we designed an experiment to address the question, how does this multivariate thermal phenotype respond via plasticity or adaptation to variation in the oxygen environment? To do this, we measured a number of traits in lizards, including the critical thermal maximum, the critical thermal minimum, preferred temperatures, resting and maximal metabolic rates, running endurance, as well as hematocrit and hemoglobin. Now, I mentioned this project was a fruitful collaboration amongst us researchers, but also with the lizards involved, who were only too happy to participate. And you can see examples of that here and here. So we measured all of these traits on lizards collected from populations at low and high elevations within their current range. We brought these lizards into the lab and acclimated them for 3 days, and then measured all of these traits. We then kept the lizards in the lab for an additional 3 weeks at their elevation of origin, and split each group in half, moving half of each group up to extreme high elevation. So this is the Pic de Midi research station at about 3,000 meters above sea level. This is above the current range limit of the species, but within an area they could plausibly colonize as temperatures continue to increase in the coming decades. This experimental design allows us to test for the relative effects of local adaptation in lizards originating from low and high elevations, as well as for the effects of plasticity in response to this novel hypoxic environment. To analyze these multivariate thermal phenotypes, we utilized multivariate statistical tools, specifically the RRPP package and R. So what I'll show you here is the mean for each group as well as a 95% confidence ellipses projected onto the first 2 axes of variation. You can see principal component 1 on the x-axis representing 46% of the variation. And here strongly loading are the traits, the thermal preferences, hematocrit, hemoglobin concentration, and running endurance. On the y-axis, we find principal component axis 2 representing 26% of the variation. And here we see strongly loaded the critical thermal minimum as well as resting and maximal metabolic rates. So I'll show you our first group here. This is lizards measured at their elevation of origin after 3 days in captivity, and then after 3 weeks in captivity. Notice here we see some extreme shifts in the thermal phenotype in response to this time in captivity. Specifically, they're shifting along principal component 1, And these changes include reduced running endurance as well as decreased hematocrit and hemoglobin. Now, this is important because often studies such as this are conducted with single measures of individuals at a single time point. And we show here that the amount of time they spend in captivity, even at their native elevation, can dramatically change their thermal phenotype. Additionally, notice here that these changes are parallel, that lizards from low and high elevations are changing in response to the time in captivity in the same direction. Next, I'll show you our results from the second time point, comparing lizards kept at their elevation of origin to those transplanted to extreme high elevation. And here again, we see dramatic shifts in the thermal phenotype. Notice that these shifts again are parallel, and notice that the shift is much more extreme for lizards originating from low elevations. And this is exactly what we would expect. This movement up to extreme high elevation represents a much more dramatic change in the oxygen environment for these lizards, and so we expect that their thermal phenotype changes that much more in response. And finally, I'll show all of our results at once to illustrate one final pattern, and that is a rather clean partitioning along the second PC axis of lizards originating from low and high elevations. And we interpret this to represent local adaptation. Regardless of testing conditions, lizards originating from low and high elevations differ in these traits, specifically the critical thermal minimum, as well as resting and maximal metabolic rates. And this fits with previous reports in the literature showing local adaptation along an elevational gradient in these traits. With this in mind, we interpret the first axis of variation to represent plasticity. These are traits that shift in response to captivity and in response to the novel oxygen environment. Our current work is being directed towards understanding how these processes can interact and ultimately shape, either facilitate or constrain the ability of these lizards to continue colonizing higher elevation habitats. I'm looking forward to continuing that work and would like to thank a number of funding sources for this project, as well as others who have supported us. Please feel free to contact either Brooke or I with questions. You see our email addresses and our Twitter handles here. We would be happy to discuss this in more detail. Thank you very much.